extend a quick thanks to the organizers for this opportunity. It's a real honor to share my work with you all today. You may notice that the title of this presentation is a little different than what's in the agenda, and it's because I felt for this shorter talk format, it was better suited to tell just a different side story that's come out of our transcription start site sequencing in Zymomonas. So I think it goes without saying that in order to meet the rising energy demands of our society, we need to develop renewable and sustainable alternatives to petroleum-derived products. And at the Great Lakes Bioenergy Research Center, we're working to develop microbial platforms like Zymomonas mobilis for the conversion of plant biomass into specialty biofuels. In order to do this, we need to rationally engineer these microbes to not only metabolize what are often non-native carbon substrates, but then to divert that carbon flux away from native pathways and into synthetic ones that produce our desired products. What makes Zymomonas particularly attractive for this purpose largely stems from its fast and highly productive conversion of glucose into ethanol via the Entner-Duderoff or ED glycolysis pathway, which is not only more thermodynamically favorable than canonical EMP glycolysis, but also generates less cellular biomass and allows Zymomonas to produce comparable yields of ethanol using 1/5 the cellular resources that would be required in either yeast or E. coli. What's more, Zymomonas is more robust against the inhibitory growth effects of the lignocellulosic hydrolysate-derived toxins. So you compare the impact, the reduced growth rate in yeast, in the presence of these so-called LTs, compared to Zymomonas. And so it's these and other attributes that make Zymomonas an efficient and robust platform for bioethanol production. But we want to leverage these attributes to make something other than ethanol. And to do that, we first need to address critical knowledge gaps in our understanding of this organism. To that end, we recently resequenced the genome of Zymomonas mobilis strain ZM4. This is the parental strain of a number of xylose and arabinose engineered metabolizing strains that we work with. And from this work, we will for the first time be reporting on the sequences of 4 native plasmids along with the corresponding protein coding gene annotations, or predictions, which brings the total number of predicted protein coding genes in Zymomonas to about 2,000. So this is a relatively small genome that we're working with, right about 2.2 megabases. And to build on this work, I'm collecting a number of genome scale data sets to further characterize the Zymomonas transcriptome to address key aspects which we're sorely lacking in, such as complete and detailed delineation of transcription units, as well as functional gene annotations, identification of promoters and regulatory motif elements, as well as the identification of regulatory factors, mechanisms, and networks. To do this, in part, I'm leveraging 2 sequencing methods known as transcription start site sequencing and termination sequencing, which selectively target and sequence RNA 5' and 3' ends respectively. And with the single nucleotide precision of these techniques, it becomes relatively straightforward to elucidate both promoter and terminator sequences because we know exactly where we need to be looking. And this is particularly important because it's these 2 regulatory elements that govern most of the transcriptional activity in bacteria. What's more, a recent study actually demonstrated that integration of these data with traditional RNA sequencing could be used to globally identify instances of complex riboregulatory elements like attenuators and riboswitches. Yeah, okay. So generating these libraries is relatively straightforward, but there's a few key things that you need to know about transcription start site sequencing, and since this is the focus of the presentation, I want to walk you through just a couple of important details. Okay, so unlike eukaryotes, bacterial mRNAs are not capped. Instead, there's just a triphosphoryl group on the 5' end of the RNA. And the first thing we need to do is to convert this to a monophosphoryl group in order to allow for sequencing adapter ligation. Now, historically, this has been achieved using tobacco acid pyrophosphatase, or TAP. Some of you may know that this reagent is no longer commercially available, so we've instead adapted RNA 5' pyrophosphohydrolase, or RppH, for the same purpose. Once you treat the RNA with RppH and block 3' ends, you can directly ligate your sequencing adapter, which would direct sequencing of your final library fragments in the direction shown on the slide, such that the first base of your read corresponds to the first base in that RNA, and all downstream analyses focus on that very first base of your read. What may not be immediately obvious in this example, though, is that once you treat your RNA with RppH, there's nothing to distinguish true transcription start sites from the existing RNA monophosphoryl RNAs that are also present in your cell, along with a variety of other RNA 5' species. So these monophosphoryl RNAs are generated from both endonucleolytic processing events as well as intermediates in mRNA degradation. So we need a way to distinguish between these two populations of RNAs. And there's a couple different ways to do this, but for this particular protocol that we're working with, we actually generate libraries from RNA that has been treated with RppH as well as RNA that has not been treated with RppH. And this way, we're able to actually to assess that background of 5' monophosphoryl RNAs in our sample. And then when we try to identify transcription start sites, we look for an enrichment in signal specific to the RppH treated RNA, as we see in this example here. So we applied this method to Zymomonas and preliminarily identified 1,000 transcription start sites. This example I'm showing you here is just a nice overview of what this data looks like alongside traditional stranded RNA-seq data as a reference. What you do not see in this example, though, are the various instances of transcription start sites indicating unannotated transcription, antisense transcription, as well as even same-sense overlapping transcription that appears to be occurring in Zymomonas. All right, so I just want to share with you one example today of how this data is impacting our ability to engineer specialty biofuel synthesis in Zymomonas. And this comes from the work of a grad student in the lab by the name of Yang Liu, who's working to divert carbon flux in Zymomonas by targeted gene repression. And to understand why we need to do this, I need to tell you a bit more about ethanol fermentation in Zymomonas. Okay, so what you need to know about Zymomonas is that the Entner-Duderoff pathway metabolizes glucose into pyruvate, which is then acted on by pyruvate decarboxylase, or PDC, to generate acetaldehyde, which is then further reduced into ethanol. And as I'm trying to emphasize in this schematic, it's this pyruvate decarboxylation step that is the primary driver of carbon flux into ethanol. And in fact, PDC is one of the highest expressed genes in Zymomonas, and this single enzyme makes up as much as 5% of the Zymomonas proteome. This is great if you want to make ethanol, but the problem is that, and problem in this instance is that pyruvate is a key precursor for the synthesis of other specialty fuels. So in order to generate these products in appreciable quantities, we need to shut down this branch of the pathway by disrupting PDC activity. Unfortunately, we have not been able, ourselves nor others, have been able to generate a full knockout mutant of PDC, and we suspect this is due to some sort of essentiality of the PDC gene for Zymomonas. So instead, we've sought to repress or knock down expression of PDC, and all while doing this without the use of CRISPR technologies, which we're still working to get up and running in Zymomonas. So what Yang ended up doing was to design a transcription activation-like effector protein, or tail protein, that would recognize the promoter of PDC, which was actually first identified and characterized back in 1987 by primer extension assays and was validated by our transcription start site data. And the way that these proteins work is that when they're expressed in the cell, they recognize a DNA sequence, and they bind to that region, and then they would prevent RNA polymerase from initiating transcription and effectively repressing expression of the downstream gene. So Yang designed this tail protein and put together a GFP expression construct to test out the activity of this tail protein. So he had GFP driven on the expression of the the PDC promoter and then an IPTG-inducible tail protein. And in the presence of IPTG, he observed a 97% reduction in GFP fluorescence, even though without IPTG, we still see a marked decrease in GFP fluorescence, and we think that this is due to low levels of leaky expression of the tail protein in this construct. So we then moved on to expressing just the tail protein in Zymomonas and assayed PDC activity in cell lysates. And what he observed upon IPTG induction was an approximate 60% reduction in PDC activity, which is still really good, but it's not quite on par with that 97% reduction we saw with the GFP fluorescence. And simply put, Yang was not satisfied with this result, and he began hypothesizing reasons as to why we were getting this particular result. And one of the things he was concerned about is the susceptibility of tail proteins to be removed from the DNA by actively elongating RNA polymerase. And so he thought maybe there's a gene, an unannotated gene that's transcribed immediately upstream of PDC, or perhaps even another promoter. And at this time, we had just gotten in our preliminary transcription start site data, so he came to me, and sure enough, when we looked at the data, we saw another 500 base pairs further upstream, a very prominent transcription start site, which corresponds to an unannotated region of the genome. But our matching RNA-seq data actually indicates that this distal promoter produces a 5' elongated PDC mRNA, which raises a number of questions about PDC gene expression regulation, right? So having identified this second promoter, Yang designed another tail protein, which when co-expressed with the first tail protein, he was able to then reduce PDC activity by greater than 70%, which means that we are now able, for the first time in Zymomonas, to reduce or to divert the vast majority of carbon flux away from ethanol and into specialty biofuel synthesis. Thank you.